Hi APE students, welcome to AP Environmental Science video 11. It is on evolution. So after this video, you should be able to describe how natural selection leads to evolution, discuss how an organism's environment can contribute to its evolution, and be able to distinguish between convergent and divergent evolution. So over the 4.7 billion years that the Earth has been here, we had about 1 billion years of chemical evolution within the Earth itself. So you had things forming like the inner core, outer core, mantle, and crust because of density differences. Once you have that basic distribution, then you can start to have small organic molecules forming, which will then lead to single cell eukaryotes and prokaryotes, and we can get the variety that we see down here. So basically, the history of the Earth is about 1 billion years of chemical evolution before we ever see any kinds of life. And then we have had about 3.7 billion years of biological evolution. So these are two terms that you should be familiar with from biology. Evolution, you've probably heard a lot, is change over time. A species changes over time through changes in genetic characteristics. So you can get simple gene variability between different species. You can also get mutations that occur randomly. And then these things have to be able to be inherited. Most of the time, mutations actually harm an individual, but sometimes they can be mutations that are beneficial to a population. And then natural selection is working on individuals, with those individuals that have certain traits that are more likely to reproduce than the individuals that don't have those traits. So again, the distinction here is natural selection happens to individuals, which then will lead to evolution of a species over time. So you should have a basic understanding of Charles Darwin. He was a British naturalist, and his big thing was going out and surveying, mapping the world. They went all over the world on this voyage of the HMS Beagle. You'll see that that was about 5 years. And then a little over 20 years later is finally when he published his book on the origin of species. And you probably are aware that people did not really take him seriously at the time. They called him Monkey Man because of his theory of evolution. But now this is the scientific consensus in biology, and it's kind of the backbone of biology. So here's his voyage. You'll see the red line going all across the Earth. This yellow dot is the Galapagos Islands. He studied finches there. And what he noticed is that on South America, there was only one finch. And then in the Galapagos Island, there were 13, which was kind of curious to him because if there's only one on the mainland, you would assume that there would only be one on the islands. So looking a little bit more closely at these finches, again, here's the 13, two of the 13 that were on the Galapagos Islands, and you would think that there would only be one if it came from South America, which it did. So the differences in the finches were the beaks, and Darwin proposed that it was because of adaptations to a new environment. So in other words, the food on the island was different from the food in South America, and so the finches on the island had to adapt to that difference in food. So what he hypothesized was that one finch at some point in time went from South American continent and got to the island, and then that one finch diverged into those 13 different species. So what we're talking about here is speciation. One species splits into many, so that one species of finch led to the 13. It's also really important that you understand that this is what divergent evolution and adaptive radiation are. So these terms can be interchangeable. Speciation is the same thing as divergent evolution, which is the same thing as adaptive radiation. And usually the way you'll see this in a diagram is you have one organism and it's branching off into a bunch of different species. And that's how evolution is occurring. So how can these different things take place? Well, one way we can get speciation is by tectonic plates shifting over time. So you'll probably notice, you probably know about Pangea is when all the continents were together 225 million years ago, also the time of the dinosaurs. And then as you go through these diagrams, you'll notice that the plates are splitting apart. They're doing this because of continental drift and plate tectonics. The Earth's mantle is moving and causing the plates to shift around. And when that happens, you actually end up getting spaces in between the continents that allows or does not allow the species to move around anymore. So if I'm an animal over here in Pangea, I can walk all over the place. I can go anywhere. And then present day, I cannot easily get from Africa to North America. So that's going to separate me and cause me to become a different species if I'm one type of organism on Africa and the same type was on North America. They got split. Earthquakes and volcanoes are linked to plate tectonics, which we'll talk about when we talk about geology. But earthquakes can cause plates to shift more quickly, which can separate organisms, and then volcanoes can actually form new land, can form islands, and that can be how you get things like the speciation from Galapagos finches if a new island occurs. So climate change can also affect and impact organisms. Different climate zones have shifted across the planet. We actually have, for example, fossilized ferns in Antarctica that we see, and we know now that Antarctica is very cold, ferns will not be able to grow there, but at one point that tells us that that area was closer to the equator. Same thing here, you have a lot of ice down here in this picture, not as much ice over here, so you're going to have different organisms that can live in the environment on this side as opposed to on this side. So again, speciation, how does it occur? So geographic isolation has kind of been what we're talking about with plate tectonics, and that leads to our reproductive isolation. So if you have a species of bunny and something happens and there's a river now that has separated some of the bunnies, on one side of the river, they're going to adapt differently than the ones on the other side, and eventually you will actually lead so much to differences that if you put them back together, these two bunny species would not reproduce. So you have created a whole different species. So that's the difference there. If the species can still reproduce, then they would not be a new species. So this is how you get speciation. So kind of the opposite of that is convergent evolution. This is when different species in different places become more similar, usually because of their environmental pressures. So for example, falcon, bat, pterodactyl, obviously living in different times, but they all adapted the same kind of flying mechanism. Same thing for dolphins and sharks. They don't necessarily live in the same area, different types of sharks and dolphins, but they have adapted in the water, and they have adapted that same kind of aerodynamic body style. So looking at a couple different types of natural selection, there are three, and we're going to use kind of the peppered moths as an example. So stabilizing selection means that the average is favored. So here's your bell curve. This is how we talk about averages in statistics. So it looks like kind of this bluish color is favored in this situation. That's where most of them are. You also have white on one side, and you have like darker blue or black on the other side, and these are not so favored. So the middle is kind of favored. Now do make sure that you can match these graphs together with what type of selection it is. Hint, hint. So directional is one of the extremes is favored. So for example, on this, if we're continuing the moths, the yellow line would probably be talking about the darker colored moth. So there's way more darker colored moths and not very many white moths. So the darker colored ones are favored. And then in disruptive, you have both extremes. So you have white is favored and navy is favored, but that lighter blue is not. So either one of those is more desirable. So a really good example of this is the case of the peppered moths. Before the Industrial Revolution, this is what trees looked like over here, very light. And so if you were a dark colored moth, kind of sucked for you. You were going to be seen and eaten by bugs, birds, all that sort of stuff. And you can barely see this light colored moth down here. But it is there, it's camouflage, so it's really good to be a white moth before the Industrial Revolution. After the Industrial Revolution, you see this dark tree over here because we were burning coal and we were putting so much ash into the air, it actually made the trees darken with pollution. And so now it's more desirable to be a brown moth than it is to be a white moth. So this is a directional selection. We shifted from more desirable to be light gray towards to be the darker colored. So looking at islands, we've talked a little bit about islands already, but typically the closer an island is and the larger it is, the better diversity you're going to have, the more diversity. So in this picture down here, you have looking at distance, closer islands are going to have more organisms on them than farther away. Kind of makes sense. It's harder to get to farther away. When you look at the size, you're looking at larger islands are going to have more biodiversity. It's easier to get to a large object than it is to a small one in the ocean. And then ideally, your ideal situation would be a large, close island. that one's going to have much higher diversity. Also, looking at extinction, if you're a smaller island, you're going to have higher incidence of extinction because there's less diversity than if you're a bigger island. You're not going to have as much extinction happening. So looking at a couple of different ways that natural selection can happen, we have genetic resistance, which kind of comes from mutations. We'll talk about why you should always take off your antibiotics in just a second. So pretty much how this works is you have a group of bacteria, the red bacteria, that are exposed to an antibiotic. And these blue bacteria down here are, for some reason, resistant from mutations or something like that. When you take an antibiotic, most of the bacteria are going to die off, except you have these resistant ones left over. And then now those resistant ones are going to multiply so that you have majority resistant, and that's what you see down here in this case. So now if you have to take another antibiotic, that same one that you took to get rid of the red one, the red ones is not going to get rid of the blue. So now we have to create a whole new antibiotic. And how does this happen? Well, some ways that this could happen is when you go to the doctor and the doctor tells you to take your antibiotics for 10 days, a lot of people stop taking them around day 4 or 5 when they feel better. And what they're doing is actually creating like these blue ones down here that are going to be resistant. They've been exposed a little bit to it and they survived. And now if they're exposed to it later, they're used to it. They have a mechanism to deal with it. So please make sure you always finish out your antibiotics. So continuing on the idea of genetic engineering, what we have always done in the past is artificial selection by selective breeding and crossbreeding. This is how we have all the different types of dogs we have. We literally just took different dogs with different characteristics and put them together. And then now the big thing is GMOs, genetic engineered organisms, genetically modified organisms. So an example down here is this rat that's a normal size. We put in a growth hormone and made it twice as big. So that would be an example of a genetically modified organism. And looking at the process of how does this actually work, really you should just be able to understand this first phase up here. So you have DNA from some organism, you can cut it and take the parts of it that you want, which is this little red piece right here, and you can actually put it into another organism. And then once you've done that, you are taking whatever gene you wanted from the other organism and putting it into another one. So this is kind of the basic idea behind genetic engineering. Now there is some controversies over this, as you can probably imagine. There's some ethical issues about, what kinds of things are we creating? Do we really know if there are any harmful implications of it? There's also things talking about kind of looking at designer babies and seeing if we can, make the best features. Oh, I want a baby with blue eyes or brown eyes. I could potentially be able to pick things like that. So that's kind of your ethics and looking at privacy issues. You also have legal and environmental issues. We don't really know what some of these organisms could do to the environment or if they could have any negative implications, just because it's an area that's uncharted. We don't really know anything about it. Although we do know some things. We do know about the ones that we are actually creating right now. And we'll talk a little bit more about GMOs with food when we get there. Okay, so bring your questions in tomorrow and be ready for quiz.